soil. It was not in the soil. It was anything in the soil. In horticulture.
Please explain.
What is this here? Dr. Kannan is there. If he wants, he can present, or only. He's the content author, so he can present it.
Sir?
Dr. Abhishek? Abhishek?
Yes, ma'am.
I think...
ma'am, actually, they all belong to a same lab and they must be facing some internet issue.
The same issue they are facing.
Sir? Good afternoon, sir. Sir, yes, the session was planned yesterday, sir. So we are planned, we are ready at yesterday to 3:30 to 5.
Yeah.
So yesterday was finished. So today we are not expected this time, sir. So suddenly sir told to attend, sir. So network connection was very poor, sir. We are facing network problems. So that's why only not. Sorry.
It's okay. It's okay. We understand the issues related with the internet and it's beyond our control. So, but we are here. And are you ready to share your slides?
Yes, sir.
Yeah, please,
yes, sir.
please go ahead.
Yes. Sir, it's visible, sir.
Yes, Ajay. Very good. Very nice.
Yes, sir. Yes, sir. Shall I press? Let's.
Yeah, please.
Good afternoon, all. Thank you for giving the opportunity. to present here. I am Ajay Kalapina, a research scholar under the guidance of Dr. R. Kannan, Department of Agriculture, Kalasalinganam, Academy of Research and Education. My research topic is effect of Excel powder for both performance of okra plant. First of all, introduction about 250,000 of Excel waste is produced annually worldwide. Most of the Excel waste are currently completed on site without any pre-treatment and pollute the environment. In recent years, the great deals of efforts have been performed for transforming the Excel waste to a valuable product. A single Excel contains 2.09 gram of calcium. It will support the plant growth, promotion, regulates many metabolic process and biochemical function. This Excel fertilizers made from crushed egg-cells, which are high in calcium carbonate, a crucial mineral for strengthening a plant cell wall. Therefore, the egg-cells are an effective and inexpensive fertilizer for outdoor garden soil and household plants. And this diagram, first diagram illustrates the preparation process of Excel powder. Generally, people consuming after using the eggs, the cells are getting wasted, thrown away, and the collection of pixels then purify and then grind it. Sir?
Yeah.
Ajay, explain.
Ajay,
Please share your screen.
Yes, sir.
Ajay, no. We have seen your slides, and you have used eggshell powder as a growth promoting
Yes, sir, career material.
ingredient, right? Okay, so my question is, what was the sieve size of your powder?
0.7 and 0.6 and 0.5, sir. Same
0.7 to 0.5,
sir.
okay. And eggshell is a source of calcium carbonate, right?
Yes, sir.
So do you think that eggshell alone could be a very good material for okra production, or would you like to mix it with some nitrogen rich, phosphorus rich compound? What are your thoughts about it? Usually when we take organic material, they are a mix of so many nutrients, but eggshell is like purely like calcium source, right?
Yes, sir. Yes, sir. That promotes the cell walls, plant cell walls, sir. So with the adding and when we compost to red soil, so
what about the nutrient composition of other nutrients present in this egg cell? How much?
I think it's largely calcium only.
It's largely calcium will not.
Calcium, yeah, that was my apprehension.
But calcium will like disrupt the absorption for other nutrients.
So
Madam, in South India,
excess of calcium will cause the disruption of the absorption of other nutrients. It will form the complex with the other nutrients.
because in South India, many persons give aerobic rice. They are using this calcium silicate and they found very good results.
But silica itself could also be the factor, na, sir?
No.
This is calcium carbonate, sir.
This is calcium carbonate. Okay, Ajay. Thank you so much.
Sir. Okay, sir. Sir, okay.
This can be used as one of the components in the compost or something as one of the components.
You can think of some admixture where eggshell powder plus something of based on the availability of your area, right?
And in hydroponic,
Yes, sir.
But this cannot be used for hydroponic also because this is powder. How do you dissolve in water? Do you dissolve it? Were you able to dissolve it in water? Because we need liquid fertilizer for the hydroponic and aeroponic, not the solid fertilizers.
Liquid form of fertilizer.
Hello?
They have added in okra crop.
But okay, not
Okay.
in hydroponic. Okay.
No. He has
Okay.
used in this okra crop.
Okay. Then it must be with some vermicompost or some compost. And then
Combination of fertilizers.
So we have got Dipendra Singh Yadav.
Which session?
Let me ask him only. Dipendra? Dipendra, can you hear me?
Excuse me, sir.
Yes, Divya.
Sir, sir, here is the network issue, sir.
Divya, can you just show me? Can you just show your slide and we'll ask. Just
fully present my slides, sir. Sir,
yes, Divya.
sir, now audible, sir. My voice, sir.
Yeah, very audible. Okay, now I could see Bhavna has. Bhavna.
Kusum Kumari.
Kusum Kumari.
Mm.
Bhavna Mamgain. Dipendra Singh.
Ma'am, I'm starting.
Yeah, please. So your session is 5, right?
Yes, sir.
5.3. Sir, it's 5.3.
Okay.
P 5.3. Yes, Bhavna, you can share your slides.
Okay, sir. Sir, my slides are visible. Can you see me?
Yes, please. Go on the full screen mode.
Okay, sir. Good afternoon, everyone. Myself, Bhavna. I am a PhD student from Govind Balla Pant Agriculture University. My topic of the presentation is aeroponic in vegetable production, present and future prospect. This is the content. Here I begin. What is aeroponic? Aeroponic is the process of growing plant in a air or mist environment without a use of soil or an aggregate medium. As you can see in this picture, we provide a nutrient solution to the plant root. Aeroponic is a soilless culture. The aeroponic system is probably the most high-tech type soilless agriculture system. In aeroponic system, the root hang in air or in a mist with a nutrient solution. The misting are usually done every few minutes. in aeroponic, growing media is primarily air. Now need of aeroponic. We need aeroponic for round the air cultivation. Round the air cultivation means we are cultivating plant in a controlled environment where we control all temperature and environmental conditions for better growth of plant. Plant grow faster in aeroponic system. Is a system of maintenance, less need of nutrient and water. Aeroponic system require little space and high yield. In aeroponic system, no transplanting shock is occur. Easy fruit harvesting occur in aeroponic. Disease-free production occur in aeroponic system. As you can see in this system, in this picture, aeroponic system occur in horizontal as well as in vertical. Objectives of aeroponic system: eliminate waste of vegetable during harvest caused by pathogen. Increase profit, allow for the reinvestment and expansion. Aeroponic can be applied on small, medium, and industrial large scale. Aeroponic reduce cost of maintenance required, such as fertilizer, chemical, fumigation, soil, staff, and more. Types of aeroponic. The basic principle of aeroponics is to grow plants suspended in closed or a semi-closed environment by spraying the plant dangling root and lower stem with an atomized or spray nutrient-rich water solution. There are three types of aeroponic system: low pressure unit, high pressure unit, and commercial unit. This is as you can see in this picture, this is the development of potato, mini tuber potato in aeroponic system. Firstly, plants are, as you can see in this picture, plants are in aeroponic block. This is the root development in aeroponic system. And in third picture, you can see mini tuber development start in aeroponic system, and these are the mini tubers. Now, future prospect. Soilless culture considered as a new development technique for agriculture development, but it is not simple technique. The system is an need to take strong care and observation for the parameter need for good plants, such as nutrient concentration. Now, conclusion. Aeroponic growing allow plant and crop to grow without the use of pesticide, and thus it will be pathogen free. Thus it will be decreased disease free. The crop will grow in a natural, healthy manner, as the aeroponic system is very similar to natural environment condition. Thank you.
Thank you so much, Bhavna. It was a very nice presentation. Ma'am.
Abhishek, Rania has also joined.
Yeah. So Bhavana, what again the same question, like what is your thought about the economics of aeroponic? Is it feasible with the Indian farmers?
Yes, sir, it is a visible. Sir, if Indian farmers do it, first initial cost is high, but if they once do it, they do it. If they do it properly, they start earning. Even they start earning.
Then why it is not popular?
There is a farmer which name is.
Are you doing aeroponic in your lab? Have you, or it is just a review you have just presented, or somebody in your lab is doing on aeroponics?
It is a review, that's a present and future.
Possibly if some senior or supervisor, like I would just want to get it. What is the initial cost? Like, are you aware of all this? I think we lost her.
Please log out.
All right. So now Rania, I could see Rania ID. Rania?
ID Abdullah. Rania?
Hello.
Yeah, you are audible. You are from which session?
I am from session
Fifth,
7.
seventh.
Yes.
Okay.
Climate smarts.
I have a poster.
Poster number. You remember your poster number?
17.
17. Yes, yes. Okay, please share yourself. Sir, it's 17, 7.17.
Yes.
Okay, welcome, Rania. Please share your slide.
Thank you.
Abhishek, record this session on cloud.
If I after few minutes
It's recording,
then
madam.
Okay,
with my phone I can't. I should open the PC.
IT, please record the session.
It's been recorded.
Recording, madam.
It's recorded.
Okay.
Yeah, Rania, you were saying something?
Share your screen, Rania.
If I stop now, then I will open the PC. Wait.
Yeah, please.
We open.
Okay,
We
no
open.
issues.
Okay, you see the slide?
Not yet.
It's clear? Not yet.
Rania, please share your screen.
I can't know. Wait. Rania.
Abhishek, sir.
Yes.
Shall we call another participant, sir?
Yeah, sure, but I couldn't see anyone.
Sir?
Yeah,
Sir, shall I share my screen, sir?
yeah, sure.
Thank you, sir.
Wait. Somebody has shared. Okay, Rania shared. After that, Divya, you share your screen. Okay.
Okay, sir. Okay, sir.
Hello.
Yes, please.
So I can present the poster.
Please mute one of your mobile.
Okay, is that the stop the mobile now?
Yeah.
So thank you. I will present our research study of about microbiome associated with tomato and trees. This study is a part of our laboratory research, production and protection for sustainable horticulture, and part of Korea Africa Food and Agriculture Cooperation Initiative project activities. Plant-associated microbiome has been known to be one of the key determinants of plant health and productivity for more than a century. They confer fitness advantages to the plant host, including growth promotion, stress tolerance, and resistance to pathogens. The distribution of soil microbial community was highly affected by various environmental factors, agricultural management, soil characteristics, and plant species interaction. Within each species, plant genotype may also affect rhizosperma community composition. These effects are likely attributed to the difference in the root cell component and to the chemical composition of their exudates. The interaction between soil microbial structure and genotype of the same plant species is a key factor for plant breeders aiming to incorporate associated microbiome in their breeding plan. Focus will be on complementary breeding strategy based on genotype allobionts, which microbes are one of the direct targets of the selection process that help achieve a desired plant phenotype. The current study is the first step to gain deeper understanding of an eventual link between some tomato and trees and their associated soil microorganisms. This complementary plant breeding approach is a novel technology that will help reaching novel phenotype. In this study, 15 tomato entries were explored and compared to two commercial tomato varieties, Sahel and Sankara. The composite soil sample from each individual plot were collected 4 times, before tomato planting, at 3 and 9 months post-planting, and at the end of the experiment. The soil microbial community structure was estimated using soil dilution plating techniques on various agar media. After culture incubation, the soil microbial population count was estimated as the log colony forming unit per 1 gram of fresh soil. As a result, the bacterial colony counts were more abundant at 3 months post-planting than at 9 months post-planting for all entries. And where a significant improvement was recorded from the rhizosphere soil of 5 tomato entries compared to Sahel and Sankara used as tomato cultivar controls, as shown in the diagram. Actinomycete population was higher at 9 months post-planting than at 3, where a significant increment was achieved using 3 tomato entries compared to 2 tomato cultivars. For fungi population, were similar as control, except for 2 tomato entries, which was lower than that of 2 tomato cultivars controls. In this study, 4 tomato entries are selected and will be tested for their resistance to soil-borne pathogen, were grown in naturally soil-infested soils. The obtained results clearly demonstrated that the distribution of bacterial actinomycete population varies significantly depending on tested genotype and their development stage. This variation may be due to the variation of amount and chemical composition of their root exudates. Understanding the vast microbial diversity in the rhizosphere and the interaction between microbiota and the tomato trees will facilitate the development of future strategies for plant growth promotion and protection breeding programs. So integrated the knowledge on multifunctional interaction between tomato crop production and disease severity parameters and soil microbial community in selected tomato trees will eventually lead to a sustainable solution to reduce the threat imposed by soil-borne pathogen. And thank you for your attention.
Thank you, Rania, for such a wonderful presentation.
Thank you.
So when you say microbiome, we initially think of all the living community in the rhizosphere, but what I could see is it's the cultivable microbes which you have focused in your research, right?
Yes.
Or are you planning to go for high throughput sequencing also?
Yes, now just culturable microbial, and this is the first essay this is the first study, but after that we will collaborate with other lab to do through sequencing analysis.
That would be
because it costs many.
Yeah, but then you cannot say on the basis of these that you have understood the microbiome, isn't it?
What?
On the basis of plate dilution experiments, you cannot claim that it is a rhizosphere microbiome.
Yes.
All right. Acha, did you identify some potential microbes from actinomycetes or tomato, or you are just
for this study just to evaluate the variation between the tomato rhizosphere, but in a previous study we have identified some potential plant growth promoting or antifungal bacteria, also actinomycete from compost for their ability to control tomato diseases such as fusarium and sclerosium.
Okay, good.
Thank you.
Thank you, sir. Divya?
Yes, sir. Sir,
Yeah,
I share.
yeah, please.
So now visible, sir.
Yes, it's visible. And we have heard the initial part of it. So can you just go down and explain the, just discuss your results.
Yes, sir. Is visible, sir?
Yes.
Okay, sir. So now I collected the soil sample in rhizosphere area, sir. So totally I have isolated in 32 bacterial isolates. And then only the out of 32 isolates, the 7 isolates is performed the best result of in PGPR activities, sir. So the bacterial isolates name is 523, 28, 32, 34, 37, 38. These are isolates is best performance in PGPR activity. And plant group promoting activities such as potassium and phosphorus and siderospore production and hydrogen cyanide production. Then I studied the vigor index in the crop of rice, the variety of ASD16. The 7 potential isolates is best performance of in vigor index activity, such as root length, shoot length, root dry weight, shoot dry weight, and germination percentage. These are the 7 isolates. Then I go to the consortium mode. Then I tested the compatibility activity of these 7 isolates. The 7 isolates is performed. The 4 isolates combination of 2 products, and 3 isolates in combination of 2 products. Totally, I have 4 combination of products, sir. Then I have my future work was in molecular characterization and self study assessment and the commercialization of my product to farmers.
Yeah,
Okay.
student Divya.
Sir,
Yeah, didn't identify the bacteria or fungi? Which bacteria is that one? Genus of the bacteria.
no, sir. Now I only in the in vitro assessment of studies, sir. Then only I do the molecular characterization, sir. Next, my work is molecular characterization, sir.
Yeah, you can morphological characters by under the
Yes, sir.
microscope.
Gram staining and
Yeah, through gram staining you can
all of
detect
it.
now the shape of the bacteria.
Yes, sir.
Divya,
Sir.
first of all, I would like to give you a compliment here, or just microbial isolation, and your hand is very good in microbiology. I could see the streaking plates are very nice.
Thank
Very
you,
nicely coordinated. What is the title of your top? Can you go up? Compatibility of property of bacterial isolates, right? So
yes,
we feel like that it is a kind it is a study of the rhizospheric microorganisms, but then in conclusion you are using the word endophytes. Go to conclusion. The promising
sir,
endophyte
this is for
selected.
epiphytic, sir.
Yeah, it's wrong.
So you try to remove this discrepancy, right?
Quite mistakes.
Yes, sir. Okay, sir.
Endophytes and all those things. Okay.
Okay,
By
sir.
rest all I could see the same. This experiment table 2, it is coming from the lab experiment only, or you have
Yes,
done it
sir. Lab experiment in vitro conditions, sir.
in organic? How do you explain the compatibility?
Sir, compatibility study using the nutrient agar media, sir, and using the streak plate method, sir. And
No. Yeah, please.
So the image I shows the compatibility test, sir. So one plate I stick the 3 isolates in cross streak method. It's a compatibility in not lysis the in bacteria juncture. It is the compatibility of the bacteria, sir.
Okay, but then compatible does not always mean they are synergistic, right? They are not promoting each other's growth, but they are just living there in the same plate, right?
Yes, sir.
So they could be neutral also.
Sir?
Yeah.
Okay, fine. Thank you so much.
Thank you, sir.
Yeah, so anyone else who is in the participant list and want to share their slides?
Ajay Kallapiram.
Ajay has already done, I think.
This Srinivasa Murthy.
Srinivasa Murthy, are you there?
Srinivasa Murthy.
Sorry.
Yes, share your slide.
Yes. Sorry, sir.
Just wait a second, actually. Yes, it is visible.
Which session?
It's same, sir, 7th.
Sorry, screen is visible, sir.
Yeah,
screen is visible.
Screen is visible.
Little bit zoom.
Sir, it is 7.12, 7.12.
Okay.
Srinivasa?
Sir,
Yeah, please go ahead.
Education. I started work by collecting my soil samples from
Srinivasmurthy, your voice is not clear. Voice is breaking.
Hello, sir. Hello, sir.
Yes, Srinivasa, we have seen your slides. So, what is the difference between actinomycetes and fungi?
Sir, it's a similar structure and the filamentous of actin is very short when compared with the fungi.
How will you?
We speak in plate, agar plate means the division of filamentous in integrate to agar plate and the surface is like a powder in nature.
Okay.
And I study various PGP traits in in vitro condition, like potassium solubilization, and actinides replace it grow in very high conditions. I selected a 5 culture in that in specific PGP traits, and we transferred to making a product after a molecular classification of in vitro study.
Srinivasmurthy,
Sir?
for isolation of actinomycetes, which media you used?
I used Acting is isolation of the soil.
Sorry.
Hello, sir. It's audible, sir.
Yeah, and you have used native.
Yes, sir.
From where?
Near to Virna district of Nadikudi village.
And what was the crop?
It's a crop like wheat and also a bindi.
Wheat and brinjal, you are like mixing together, or you are isolated some from wheat rhizosphere and some from brinjal rhizosphere?
Oh, no, sir. It's a bindi recipe size. Bindi plant.
Srinivasa Murthy, how the wheat will grow in South India? Which place you collected?
Nadikudi village, sir.
Which part of South India you collected the sample, soil samples?
Apart from Virna district, sir. Virna district and Nadikudi village.
And where you are, your lab is there?
No, sir. My lab is in Kalsanga University in Krishnagiri.
So it is native. Is it native? That actinomycete is native to your lab?
No, sir. Small distance we are collected.
Okay. Okay, Srinivasa. Thank you so much and all the best.
Thank you. Thank you, sir.
लगता है आपको नजर नहीं आ रहा मेरे ख्याल में all they have presented.
I think we have taken presentation from all the participants who are appearing in the list.
Divya, Srinivasa Murthy, Nikesh also finished, sir.
Divya, Ajay.
Ayushi also finished offline.
Ayushi, would you like to give again? Sir, I think we should wait for another 5 minutes and then we'll ask ma'am if to continue or we can call off this session. Ajay, Nikesh, Srinivasa, Divya, you can log out.
Okay, sir. Thank you so much.
Oh, okay.
Thank you, sir.
Yes. Ma'am.
सभी लोगों का हो गया?
मैम जो आए थे हमने सभी का कर लिया है. अभी कोई भी पार्टिसिपेंट लिस्ट में नहीं दिख रहा है हमें, तो आप बताएं.
अभिषेक?
यस मैम. हेलो.
हो गया सब लोगों का?
हाँ जी मैम, हो गया है.
अच्छा, फिर भी 4 बजे तक उनका टाइम दिया तो बैठे रहना पड़ेगा
जी.
ठीक है, तो आएगा तो देखिए. ये है 6 से पहले 7 होता तो देख लेना. 6 लोग है ना, 6 से अगर 7 होगा तो देखिएगा.
That is, I think, if you are promoting a company like this, you must always encourage them to take this soft loan. It is as good as grant. इतना सिंपल. और ये 5% आज का नहीं है, 25 साल से चल रहा है. और उस समय में इंडस्ट्री बहुत ज्यादा थे मार्केट में. अब 20% में मिल रहा है, 25% में मिल रहा है, या 5%. इनका इंटरेस्ट रेट्स देखिए कहाँ से कहाँ है। तो 5% इज एज गुड एज आप मार्केट में भी मिलेगा तो क्या मिलेगा 12% तक मिलेगा, हालांकि अभी भी है। Let's focus on, और ये बात सही है कि हमको अभी भी पॉलिसी तो बनानी चाहिए स्टार्टअप हालांकि फाउंडर हैज बीन टॉकिंग एंड प्रमोटिंग द बिग वे बट मेनी टाइम्स डॉक्टर सलाह होती saying that I think startups have promoted startup. So given that strand, this book thinking of those terms. Rajiv, anybody else wants to participate?
I will go back to just person, and if possible, can we have a meeting recording for YouTube, sir?
Yes, of course.
So that it will be better able to tell you, sir.
Yes, yes. Dr. Ramanand, Ramanand is Kolkata. Yes, yes.
रणनीति एक तुम फोटोग्राफ पहले ऊपर से तो हर चीज के लिए बढ़िया क्यों जो करते थे बेटा